Well, good morning, everybody. Trying to figure this out, so you're going to have to bear with me. Virtual elbow bump as we get started here. Hopefully, this will work. So we'll see how it goes. What I want to do is start to talk to you guys about the content that's in chapter 23, which is what we would call population genetics. It's on the evolution of populations, and where we're going to start with chapter 23 is by taking a look at something called microevolution, which is simply just changes in the allele frequency of different alleles in a population over generations. So what's important to note here is that we're talking about changing a population over time. Individuals in a population do not evolve, they don't change. Populations evolve, populations change. So, for example, there have been studies done on finch populations in the Galapagos Islands where, in periods of drought, the seeds become really hard and tough to eat, and the finches who have the thicker, bigger bills actually have an advantage when it comes time to try and eat and find food. And so, those particular individuals have an advantage. And what we see is that in the next generation, there are more finches born with those larger, thicker beaks because their parents were the ones who were able to survive and also to reproduce. Notice that individuals that were living during the drought that had smaller beaks did not evolve or change to have bigger beaks during their lifetime. So microevolution, changing in allele frequencies in populations over many generations, can only occur if genetic variation exists in the population. And there's two ways that we actually see genetic variation occurring in nature. And generally what we see is that we see variation in phenotype, which is reflected by variations in the genotype. So the types of variations we can see quantitative variations where we see genetic variation along a continuum. And like, for example, with flower color that you see here, with skin color in humans, with height in humans, those are all quantitative characters that have variation in phenotype along a continuum, and that is reflected in their genotype, in the genotypes existing along a continuum as well. Because generally, as you hopefully remember from our previous unit, when we have these quantitative characters, they're usually inherited through polygenic inheritance, right? Many different alleles working together, many different gene pairs working together to determine a particular trait. Most variation that's heritable, in other words, that is inherited along these continuum. Most variation comes from polygenic traits. Most of our traits are polygenic. There are also what we call discrete characters, where you are either tall or short. You are either red or white, if you're a flower, for example. Those are inherited on an either or basis. And these are usually due to the actions of a single allele pair, one gene locus within the genome. So one thing that biologists, of course, will do is they will measure genetic variation within a population, and they can do that in a couple of ways. They can calculate what's called the average heterozygosity, or the gene diversity within an organism. It's been done with Drosophila melanogaster that you see in the top video or top picture right here. It's been done in zebra mussels as well that you see here. When you take a look at Drosophila melanogaster, about 14% of their genetic loci are heterozygous. The other 86% in most Drosophila melanogaster are homozygous, or what we call fixed. And then when we take a look at zebra mussels, anywhere from 27 to 43% of their loci can be heterozygous at any one time. The more variation that exists, the more there is for natural selection to act upon, and the more likely it is for allele frequencies to change. Another way to measure diversity between, or genetic variation within a population, is to measure nucleotide diversity. So you're going to compare DNA sequences of two individuals and then do that over and over and over again, comparing individual A to individual B, and then find another two individuals and another two individuals and another two individuals. And so you average all of that data from many such comparisons to figure out how diverse the genome is for any particular organism. In Drosophila melanogaster, the nucleotide diversity, the difference in DNA sequence between any two individuals is there's about a 1% difference between any two Drosophila melanogaster that you would compare. Whereas with humans, there's a 0.1% difference between any two individuals that you would choose to compare on average. So, where does variation come from? Well, the ultimate source of new alleles is, of course, mutation. Those can be point mutations. They can be chromosomal alterations. So I want to go back to this point mutation picture right here. This is from the lab of Richard Lenski at Michigan State University, who's been running what is called the long-term evolution experiment with E. coli since 1988 or 89. Not important really, but it's been going on for almost 30 years now. And so he's cultured over 60,000 generations of E. coli and been able to track genetic changes as they've occurred and go back and look at, okay, up until this point, these E. coli didn't have this ability. After this point, they did. One of the defining features of E. coli is that they're not able to digest the nutrient citrate. That's one of the reasons they get classified as E. coli. And in these cultures that you can see here, one of the nutrients in the medium is citrate. And for the first 33,000 generations, none of the E. coli were able to digest that particular nutrient. Around generation 33,000, all of a sudden in one of these flasks, the E. coli in there were actually able to start digesting citrate. So this is a novel new ability that was gained around generation 33,000. And the cool thing is then what they do with each with each of these flasks is they will freeze a little bit of the sample, and so they can go back and check and look and see, okay, at this generation we weren't able to the E. coli weren't able to digest citrate. Now all of a sudden in this generation they are. All right, let's do a genome comparison. What's different between the genome in this generation versus this generation, and pinpoint the exact genetic mutation that occurred to allow the E. coli to begin to digest citrate. Okay. Other types of mutations can be chromosomal alterations that alter the number of genes in a particular organism, either through duplications due to copying errors during mitosis, like you're going to see right here, or to deletions that might occur where a portion of a chromosome gets deleted, again, during errors of DNA replication. Gene sequences can be inverted during the course of DNA replication. You end up getting a reorganizing of a particular chromosome, so the gene sequences get inverted. And we can create new genes where you have translocations occur between non-homologous chromosomes that actually end up exchanging pieces of DNA. All of these generate new combinations of DNA, new alleles, and one of the ones I want to take a look at are duplications, which are often harmful, although sometimes they are beneficial. Our ancient mammalian ancestor originally just had one olfactory gene. Now, hopefully you know that olfactory genes, and olfaction refers to smelling. That's why I've got this picture of Jimmy Durante here with his gigantic nose, who's famous for having such a large nose. But our ancient mammalian ancestor had only one olfactory gene. However, over the course of time, that olfactory gene got duplicated over and over and over again. And actually, modern humans have over 1,000 different olfactory genes, although 60% of them are now inactivated as a result of mutations that have occurred over time. Mice have about 1,300 olfactory genes. 20% of them have been inactivated via mutations over time. So if you take a look over here at this little chart that I've got here, mice have about 1,130 olfactory receptor genes that they produce, whereas humans, we only have 396. That's about 60% of 1,000, right? We're inactivated, leaving, which would be 600. We're inactivated, leaving around 400 being active in humans. So this should tell you, of course, who is smell more important for, humans or mice? Obviously mice. And it's believed that these duplications were actually advantageous because the more olfactory receptors you have, the more you are able to pick up faint odors, which of course would be an advantage a long time ago. All right, now mutations in terms of their frequency, they occur more often in organisms that have a rapid life cycle, even if the mutation rate isn't a lot higher. For example, in prokaryotes and viruses, they have a much faster life cycle. So just because of that, you're going to have mutations occurring much more frequently. You can see down here on the x-axis, we've got the genome size. Over here on the y-axis, we have mutation rate. So higher eukaryotes, which humans would fall into this group, you can see we have large genomes and a smaller, lower mutation rate. Lower eukaryotes, this would be like Saccharomyces cerevisiae, the yeast would fall into this lower eukaryote group. They have a little bit smaller genome size, but really the mutation rate's pretty similar. Bacteria have a slightly higher mutation rate, but not really much than our eukaryotes and higher and lower eukaryotes here, but because they will go through so many generations so much more rapidly, mutations do build up in a shorter period of time within a bacterial population. And then you can see here, viruses really have a much higher mutation rate, and the frequency of mutation is much higher in organisms that have an RNA genome, like RNA viruses. And remember, we learned earlier about viroids, which are just circular pieces of RNA, have a really high mutation rate. RNA is less stable than DNA and mutates much more frequently. HIV is an RNA virus that mutates rapidly, which is why it's been so hard to develop treatments for HIV, because it keeps changing all the time. Okay. Now, there are, of course, other factors that contribute to genetic variation, such as sexual reproduction, right? That is where most genetic variation comes from in organisms that reproduce sexually. So mutations create the genetic variation, and then sexual reproduction makes sure that different combinations of those different alleles that are created are passed down to the next generation. Okay. So that is where I'm going to stop for today. We will get into the Hardy-Weinberg theorem in our next video.